Okay, I'm going to keep things as short as I can. We're on page 44. That was, the previous lesson was on the lac operon, which is going to be in a prokaryote, which is, for that instance, we're looking at E. coli bacteria. Here we're going to talk about gene control in eukaryotes. Okay, so gene control in eukaryotes, that's you and I, things that have a membrane-bound nucleus. So every cell, I've already mentioned to you, contains the same DNA. Okay, your kidney cells, going back to that example, do not want to make hair because that would just be a total interference with their function. And so we know that we have to have some kind of regulation in eukaryotes as well. Okay. Largest estimates to date when this note was made was that we have approximately 35,000 genes. I'm sure you could look that up and see that things have changed in 2020. So I'm going to keep things simple because, again, we're not sitting in a class this semester. I'm trying to reduce the amount of information I give you. This is what I want you to know this semester. Okay? How is gene expression regulated? So in eukaryotes, in you and I, there are 4 mechanisms in which we shut down genes that we don't need to make. Okay, again, one more time, why do we need to shut down genes we don't need to make? Because it's wasteful in terms of energy. And secondly, you can't have liver cells making kidney cells because it wouldn't make any sense to your organs. Okay, so what are ways that we can prevent making a gene if we don't need it? All right, so what I'm going to propose here is that we're making genes that we need. Okay, so liver cells aren't going to make kidney cells because they don't have the right repressor, et cetera, or they have the correct repressor. That's going to go forward in the correct mechanism. But how can we shut down genes that we don't need to make? So in other words, if you are not in need of mucus, okay, if you don't need to make any nasal mucus because you've got enough nasal mucus at this point, how can you control shutting down that production? And this is how you would do that, okay? We have one of 4 mechanisms. So different cells rely on different mechanisms. The first one is transcription control. So this is the most common way of regulating whether you are going to make your genetic information, okay? And in other words, don't transcribe, okay? Do not transcribe the gene at all. Okay, so in other words, do not make an mRNA, all right? No mRNA. Okay, that's the easiest thing to say. That's the easiest thing you can possibly do. Don't make the mRNA at all. All right, let's say that we've made the mRNA because different cells have different tactics. So then what you can do is you can say, well, I'm not going to process mRNA into pre-mRNA, into mature mRNA. And so this is where I'm really missing you guys in front of me because I would say, what does that mean to you guys? And somebody would tell me, that means I'm not going to cap and tail. Okay, so no capping, no tailing, all right? No splicing of introns. So the mRNA is ready, it's on hand, it's good to go. It's not going to leave the nucleus though, okay? So why is it not going to leave the nucleus? Because it hasn't been transformed into mature mRNA. All right, so that's another strategy. Another strategy is, okay, we're going to transcribe, we're going to cap and tail, we're going to put that mRNA that's ready into the nucleus. But we're not going to allow translation to occur. So we're not going to allow initiation to occur. So in other words, again, think in your mind what that could possibly mean. It means that we're not going to assemble the met tRNA. Okay, so met tRNA is not going to attach to the start codon, and we're not going to assemble the rRNA. So those 2 ribosomal units, they're not coming together. So we don't have that initiation complex, in other words, okay? So we're going to prevent that from happening. We're going to keep a mature mRNA in the nucleus, but we're not going to read it yet, all right? And then lastly, this one is going all the way back to the first unit, okay? And so I always love asking this question, and again, I'm totally missing you guys right now. Post-translational control. So we transcribed, we capped and tailed, we made a pre-mRNA into an mRNA. We even let translation occur. How can we prevent this polypeptide from becoming an active protein? And the answer is this: do not allow it to take on, and do you have the right answer in your head? The secondary, tertiary, or quaternary structure of a protein. Okay, so way back to chemistry, biochemistry in the first unit, don't allow this protein to take on its secondary, tertiary, or quaternary configuration. I'm going to use the word configuration so that you guys know what that means. Configuration means its proper shape. So in other words, at this point, we're just going to leave these amino acids stuck together with peptide bonds, but they're just polypeptides, okay? They haven't taken on their proper shape yet. All right, and that's how you and I control which genes we are using and slow down their use or speed it up, okay? We can stop things at any one of these points depending on what cell we're talking about and how much we need of that gene or that protein. Okay, have a great weekend.